starts the second week. The third week, then will be a week from today. And the exam on this unit will be Thursday, a week from Thursday. And that'll be May the 24th, so make a note of that. And the exam will cover 17, 20, and 21. Format of the exam, as I said, is multiple choice, matching, and some short answer. To be marked as Scantron sheets, or bring a soft lead pencil to do so. So a week from tomorrow, Thursday, May the 24th. Okay. In the meantime, we're going to wrap up chapter 21, and we'll actually start the next unit, which will be on blood vessels and heart, before that exam. But you should have plenty of time basically to go over it. Okay.
Will we have a review over this for the test?
Pardon me?
Will you have a review?
I don't do a formal review, but if you have questions, you'll have an opportunity to bring them up. Okay, so if you have questions, write them down and bring them up in class. And as I said, the test, not from a test bank. I don't use test banks. I make up my own tests, and it comes from what I present here in class. Okay, I don't pick out minute details from the textbook. I cover what I think is important. That's what should be tested on. So right from the notes. When I have something in bold or highlighted, it probably means I think it's fairly important. That probably means we're going to see it again, like on the test. Okay? So be prepared, and don't put it off until the night before. And then when I say there's a lot of stuff here, okay, you should be studying as you go along. Remember, each week we do is 2 weeks in the regular semester. So we have covered, or we've already covered basically 2 weeks worth of class. And we're starting actually, would normally be the third week of class in the normal semester. So keep that in mind when you're doing it. So you should now have read chapter 17 in your book, chapter 20 in your book, and I'd suggest that you read chapter 21 as well. You can study right up through the weekend up to what we finish, and we'll cover, we've already covered those two chapters, 17 and 20, and we'll be well into chapter 21 as well. Okay? Anybody have any questions on anything we've covered so far? As I said before, on chapter 17, the hardest part, I think, deals with excuse me, the blood clotting process. So make sure you understand the anticoagulants, what they are, what they do. Procoagulants we talked about. The various chemicals involved in formation of the blood clots. The pathways we talked about. Extrinsic and intrinsic pathways, which are the first part of blood clotting. And that is the formation of a very important compound called what? What's that compound? It's actually an enzyme. Prothrombin activator or prothrombinase. And that's a series of reactions. And it can be accomplished either by the extrinsic pathway or the intrinsic pathway. And what's the difference? Why do we call one extrinsic and the other intrinsic? What's the extrinsic pathway?
Outside of the blood.
We're talking about things, damage extrinsic to the blood vessels, which is frequently the case. When you get a cut or abrasion or something like that, you're usually damaging other tissues as well as breaking blood vessels. And keep that in mind. It's not extrinsic to the body, it's extrinsic to the blood. Now what starts it? What initiates it? Tissue factor, or tissue thromboplastin, which is released by damaged tissues. Okay, then you go through this series of reactions with a bunch of different clotting factors that react in a certain fashion. It all involves calcium ions. Okay? And the end result is prothrombin activator, or prothrombinase. That is the enzyme for the second phase, which is a single reaction, and that's conversion of prothrombin to thrombin. Prothrombin activator does that, or prothrombinase. Okay, and you're forming another enzyme, which is thrombin, and that catalyzes the third and final phase, which again is a single reaction, conversion of fibrinogen to fibrin. Fibrinogen, remember, is one of those plasma proteins that drifts in the blood, inactive state. When it's activated, then it forms these water-insoluble threads that form the framework or network of the clot. Okay. So that's what I mean. Think about that. And also then, when the clot forms, something else happens. The clot is a loose arrangement of cells and these fibrin threads, but you got to basically make it more compact. And that involves chemicals released by the platelets, okay? And that causes the clot to basically get tighter. And then the platelets themselves are involved in syneresis, or clot retraction, where they basically pull on those fibrin threads and squeeze the fluid, the serum, right out of the clot.
And once the clot has been formed, then the body starts to repair the blood vessel. Platelets are also involved there too by chemicals of release that stimulate fibroblast production and also smooth muscle production. And the endothelial cells, which line, remember, they get involved too. They replace the damaged epithelial cells, which are the lining. Very important. Okay? So know these different chemicals. I can't say that enough because there'll be a lot of questions on that. And if you don't, it's going to be overwhelming. And I'll try to do it the night before. Okay? Very important that you keep up. All right? So think about this. Be reading this, and if you have questions, bring them up. Okay? Before the exam. All right.
By the way, the grade book is up on PAL, so I will post the lab grades by this weekend. And then on Monday's lab, I'll have them back and you can look at them, look them over and see where you may have made a mistake or two. Okay? Or maybe I'll use yours as a key. In any case, you'll have a chance to look it over. Okay? The grades will be posted beforehand, so you can go to the PAL site of the grade book and it'll be posted, your scores. And I will do that for all exams, and often the score will be posted before you get the exam back, so you'll know how you stand. Okay? Okay. And somebody left their calculator in the lab today? Okay, we're going to look, as I said, at the immune system. We've been talking about it. I said I'll be getting into more detail soon. Well, here we go. Now, we're going to start by what is called the non-specific or innate defenses. This is what you're born with. This gives you protection against some diseases which you'll never get, okay? We don't normally get sick from plant diseases or various diseases that may affect other animals, but not us. That's not to say that some animal diseases we don't share with these other animals. Those are called zoonoses. And there are some very nasty diseases we can get from other animals. But most diseases we get from other people. So innate or non-specific defense. And when I say non-specific, that means a response directed against anything that the body considers foreign. And by now you should know the immune system can recognize foreign matter by molecules on the surface of the cells or particles. And those are called antigens. We have antigens on our own cells as well. Those are called self or autoantigens. And your immune system recognizes those and normally avoids them. You will actually produce cells that will attack your own tissues, but they're normally eliminated before they can do any damage. Sometimes things go awry, you have an autoimmune disease, and then your immune system turns on the body and starts attacking and destroying tissues. Lupus is a good example. Systemic lupus erythematosus. So is rheumatoid arthritis. So is multiple sclerosis. These are all autoimmune diseases where the body, basically, the immune system, is attacking and destroying your own tissues. But for the most part, the immune system is very efficient at separating foreign from self. And the immune response then is directed against foreign things, some of which can be very deadly, pathogens, disease-causing elements. Unfortunately, sometimes the immune system overacts and it starts to attack things, basically, that are completely harmless. And this is what we call allergies or hypersensitivities. And sometimes the response can be so dramatic it can actually cause damage to tissues. In some cases, even death. You've probably heard of anaphylactic shock. That's a very severe reaction that can occur in some people when they become sensitized to an antigen, which is usually harmless to most people. Maybe a food allergy, it may be like an insect sting or something like this, or a drug, but it can be very deadly to a person who's become sensitized to it. Like I say, for the most part, the immune system is very efficient at separating and attacking what should be destroyed and what should not be destroyed, i.e. the body tissues itself. Okay, so non-specific defenses. Now we have what are called various lines of defense. The first line of defense are the barriers. This would be the skin and the mucous membranes. As you know, the skin covers the body. The mucous membranes line body cavities that have a natural opening to the outside. So our respiratory tract, our GI tract, gastrointestinal tract, many of the tubes in our urinary and reproductive tract all lined with mucous membranes. The skin is especially good protective barrier because intact skin
Remember the epidermis, which is the upper part, it's epithelial tissue, tightly packed cells. And the stratum, excuse me, the stratum corneum, which is the upper layer, consists of maybe 20 or 30 layers of cells tightly packed, but not only that, they're dead cells, just full of keratin. So they're not much more than flattened bags of keratin. There are very few microorganisms that can actually penetrate intact skin. Usually we get infection through the skin because we got breaks in the skin. But they may just be very minute breaks because we're dealing with microorganisms, which are extremely tiny. When you take microbiology, you're going to realize how tiny they are. And I always kid my micro students, I say, you're now going to appreciate what you did in A&P when you were complaining about all those tissues you had to look at. At least you could see something, okay? Some differentiation. With microorganisms, it's mostly dots and dashes for the bacteria, and that's magnified maybe 1,000 times. They're extremely small. So it doesn't take much of a break in the skin for them to get into the body that way. So if the skin remains intact, like I say, it's a very good barrier, and very few pathogens, or pathogens of the disease-causing organism, can actually penetrate the skin. There are, of course, some exceptions. We have some parasites which are around here, which are called hookworms. They're parasitic roundworms. The adult lives in the GI tract. It latches on the intestinal mucosa, abrades it, and sucks blood. And its eggs pass out with the feces. They fall on warm, moist soil, which we have a little bit around here. Okay, the eggs will hatch into a larval stage that goes through a molt. You have an infectious larva that crawls up in the fecal matter. The person comes walking along and they step in it. Okay, the larvae will attach to the skin, burrow through the skin, find a blood vessel. The blood vessel bring them up to the lungs. They go to another molt there, then they migrate up the respiratory tree. They're swallowed, passed down the GI tract, and set up housekeeping in the GI tract, attached to the intestinal mucosa. With very heavy hookworm burdens, they can draw enough blood to actually put the person in a state of anemia. And frequently, people are infected with hookworms also have a couple other worms that they get from contaminated food and water, like Ascaris and Trichuris, which you don't need to worry about. But like I say, it's quite common in southeast United States, especially where the soil is contaminated with human fecal matter, and sometimes people are out in the pasture, well, they don't want to run back to the house, so, nobody around, so go behind the bush, and then somebody comes walking along and, you can spread the infection that way. So it is around, and it's not that uncommon, but most people are sensible enough not to, careful where they step. And you say, well, people walk around barefoot, they got this real thick skin. Well, what the worm does basically is penetrate between the toes, which is much thinner skin. They have a way to find their way into the body. So hookworms, like I say, can penetrate unbroken skin. We have another parasite, which fortunately is not common here, but it's in various subtropical and tropical areas of the world. These are called blood fleas. These are parasitic flatworms. As the name suggests, they live in the blood vessels, mostly in and around the liver. And they'll migrate up periodically. The female will lay her eggs, and normally they pass through the wall of the intestine, the large intestine, or the urinary bladder, depending on the species. And then, of course, they pass out with the feces, or they pass out with the urine. If they get into water, where you have snails, and frequently in ponds and pools you do have snails, they go through a stage in the snail, and what comes out is a little infectious stage called the cercaria. You don't need to worry about that. That swims around, and if you're in the water, bathing, swimming, or whatever, they'll attach the skin, burrow through the skin, and get into the blood, and set up housekeeping. And these things can be very dangerous because sometimes the eggs get into other organs, like the brain. It's not uncommon. So these can be very deadly. Yes?
In the news recently, a college student was swimming in a lake, and she got an open gash in her leg or something, and they had to amputate her leg. Now they're talking about the diseases up in her body. They might have to amputate both of her arms. Is that likely to be true?
I don't think so. It's probably a bacterial infection she picked up in contaminated water. When you got a wound, an open wound in the water, especially the water around here, okay, the lakes are not that clean, I'm sorry. And most lakes around the world have a disease amputation. If you're
sediments, you pick them up and they inhale the sediments, which frequently happens if you're playing in the water. It'll penetrate the mucosa, migrate up to your nerve tracts, and after your nerve tracts go into the brain, bring about primary, immediate, and encephalitis, which is fatal. And ironically, this child's parents, both doctors, his mother was an infectious disease specialist. They didn't realize what he had until too late. They thought he had meningitis, but not that he could have done anything about it anyway. This is a local lake. It's in the Lincoln Upper there. There have been a couple outbreaks in Orlando, a couple water bubbles up there. So it's around. Fortunately, it's not a very common infection, but when it occurs, it's usually fatal. It's very deadly. So if you're playing in the water, it's a good idea, you're in the sediments there, or you're picking up stuff, those bugs, those plants, and then you're, wakeboarding or something like that, and there's a possibility for difference. Like I say, it's rare, but they are here. When the water gets warm, those things get active, and then you get the sediments picked up and inhaled, you get that. But in that case, it was probably just a common bacterial infection from the water that she picked up. Got in, basically, and got spread by blood, various other issues as well. Anyway, blood flukes we don't have here in the U.S., but we do have them in subtropical and tropical areas of the world. Yes?
Did you say that the blood fluids go to the liver? Is that what you said?
Well, it usually lives in blood vessels in and around the liver.
Oh.
Okay, and then it'll migrate out, and the female will deposit her eggs, and they pass through the wall. And the problem is the host becomes sensitized to the eggs, and it becomes riddled with scar tissue. So that can shut down basically the GI tract, or at least the part of the large intestine and the urinary bladder, stuff like that. And the real danger is you can get the parasite in other organs too, and even the brain. By the way, I'm a parasitologist, if you're wondering all the spores there is. Got to throw this in here. But anyway, there is a blood fluke that we sometimes acquire. It's in the U.S., but it's not a human blood fluke, and it causes a condition known as swimmer's itch. I don't think it's common in Florida. I've not heard of it here, but the Great Lakes area, the country, it is quite common. And it's actually a blood fluke that the adult lives in waterfowl, and people sometimes, if they're in the water where the snails are, they'll pick up the larval stage. It'll migrate around and cause a very severe itching and rash, almost like poison ivy. Now, it'll eventually go away because the parasite doesn't mature, but in the meantime, you're suffering from that itching and the rash. But you get it the same way. You're waiting in the water, and you pick up the larval stage, it migrates around, eventually dies, but in the meantime, you become sensitized. That's why you get the rash and the itching as a result of it. That's called swimmer's itch. Like I said, I don't think it's around here. Has anybody heard of swimmer's itch? Probably not. Okay. But around in the Great Lakes area,
Is it the
in the Midwest, they do have it. Yes.
Isn't those contained, are those only contained to animals, or can animals contact humans?
Say again?
Can animals contact those hookworms, blood flukes?
Yes. Actually, animals, cats and dogs do have hookworms, and sometimes we can pick them up. And again, that usually doesn't go to an adult, but you may have the larvae migrated around in your skin.
Oh.
That's called cutaneous larval migrans. Don't worry about it, but it'll migrate around. Then it dies, but again, you get the rash and the itching and so on. So sometimes, yeah, you can pick it up. And especially from cats, because of course cats, if they're roaming around outside, there's no cat box, so what do they use? They use the soil. So if you're gardening or walking around with soil, sometimes you can pick it up that way too. Yes?
The same thing as what I hear people call leeches, the little black things that latch onto your skin.
Leeches?
Uh-huh.
Oh yeah,
I don't
leeches.
Know if that's a leech. I don't know if that's
No. Leeches are, yeah, they're common. No, leeches are just a blood-sucking annelid or segmented worm, but usually don't carry parasites. But yeah, they sometimes do. And you'll sometimes catch a fish, or maybe you'll see a turtle or something like that. It's got a leech attached to it. Yeah, they're quite common in the water, all over, fresh water. They scare people more than they really do any damage. You see this leech, Okay. Anyway. Now, in our skin then, we also have, of course, associated with the skin, the lacrimal apparatus, that, of course, the tear glands. And associated with that, of course, what the tears do is flush the eyeball. They flush material off the eye, including dust, because dust particles frequently carry microorganisms. We also, of course, secrete saliva. The saliva helps to wash off a lot of microorganisms from the teeth and the gums. And many cells will secrete mucus, especially cells in the mucous membranes. We'll talk more about that later. We have mucus-secreting cells. The mucus helps to flush organisms out. When you have an infection, for example, like a respiratory infection, what happens? You produce tremendous amounts of mucus because the body is trying to expel the invader. And if that invader can't attach to tissues, they're going to start flushed out. And that's why a lot of body fluids, especially mucus, when you got an infection, are contaminated with microorganisms. And it's an easy way to spread infection, as you know. That can be from the upper respiratory tract, and be something you cough off of the lungs as well. And we also have cilia on the surface of some of our cells in some of the cells that line the respiratory tract. And the cilia don't move the cells. For example, in the nasal cavity, the nasopharynx, and the trachea, bronchi, and so on, we have these cilia. And the purpose is to move the mucus-laden debris up to the throat. So you can either spit it out or swallow it, but you're cleansing the air in doing so. Remember? When you want to, oh yeah, okay. We get the respiratory tract, we're going to see that the respiratory tract is lined with pseudostratified cilia that cover up the and it's mucus secreting. So what we do is when we bring in the air, we have this mucus that traps the dust and debris, and then the cilia sweep it to the throat where we can spit it out. And then of course the urine, as you know, the urine not only carries waste products, but it also flushes out the lower urinary tract. In most cases, unless you have a UTI, urinary tract infection, your urinary tract is sterile except for the distal end of the urethra. Now, the urethra is the tube that carries urine from the bladder to the outside of the body. That, of course, is being handled. You're touching it, you got toilet paper and stuff like that, so that carries microorganisms. But what the urine does, of course, is continue to flush the urinary tract, so you're sweeping those organisms out. But you would expect that if you just took a urine sample, there'd be some bacteria in it, because there's some bacteria contaminating the distal end of the urethra. And that's why, too, when they take a urine sample, they usually want midstream to flush out a lot of those bacteria. Otherwise, you're going to have a big charge of bacteria in the first stream. So when they take urinalysis, they say take a midstream sample, put it in a little cup, and take it down to the lab, whatever. Okay, we also have some chemical factors associated with the skin. And the sebum, for example, this is the oil produced by the sebaceous glands. This helps to keep the skin soft and supple so it's less likely to crack and provide a port of entry for microorganisms. Also, the surface of the skin is acidic, usually somewhere between pH 3 and pH 5. Now remember, the pH scale goes from 0 to 14. The middle of the scale is 7, which means neither acid nor base. As you drop below 7, okay, it becomes increasingly acidic, greater hydrogen ion concentration. You can see the skin, most people, is acidic. This helps to keep down growth of microorganisms. We also secrete in our sweat lactic acid, which adds to that acidity. And sometimes you'll have something, you'll wear something, and a little piece of metal, and it'll leave kind of a dark ring. That's because the acidity in the skin is basically breaking down that metal. So if your significant other is giving you a ring or a bracelet and it leaves a dark ring, you might want to, and they told you it's gold or platinum, you might want to rethink the relationship. Maybe it's not what they said it was. Because that should not break down. But anyway. Also, too, the vagina is acidic, and this also helps to discourage growth of microorganisms.
What is the normal pH of the vagina?
Of the vagina? It varies from person to person. It also varies if a woman is douching, she can change that pH, and that's a risk. Excessive douching can bring the pH up, and that can predispose you to infections. Because by changing the pH, there are organisms that are normally found there. Some of these organisms, particularly yeast, can grow, and you can end up with a yeast infection. So it will vary, but it's usually acidic. And it's kept acidic for a very good reason, to keep down the growth of microorganisms. That also kills sperm cells. That's not the intent. And that's why sperm in the semen is alkaline, to help neutralize the acidity of the female reproductive tract. Okay, and again, as I said, perspiration helps to flush materials off the surface of the skin, and that acidity helps to keep down growth. We also have in many of our body fluids an enzyme called glycoside. what lysozyme does is it catalyzes a breakdown of a chemical complex in the cell walls of most bacteria. And that compound is called peptidoglycan. If you take microbiology, you'll go into this in some depth. But anyway, it's actually composed of simple sugars held together with small bridges of amino acids. But very important because the lysozyme, catalyzes the breakdown of that. So what it does basically is destroy or damage the cell wall in many bacteria and helps to keep down bacterial growth as a result. We find this, for example, in our saliva, we find it in our tears, we find it in nasal secretions, the mucus there, we also find it in our perspiration, and again, it's very good at helping to have an antimicrobial effect, and especially on they're called the gram-positive bacteria. And don't worry about this right now. You take micro, then we'll talk about that. But gram-positive and gram-negative, that's a major way to group bacteria depending on the composition of the cell wall. But gram-positive bacteria, their cell wall primarily is peptidoglycan. So what it'll do basically is destroy the whole cell wall. Gram-negatives have peptidoglycan, but they've also got a membrane on the outside, so it only partially disrupts gram-negative cell wall. You say, so what? Well, the bacteria have that cell wall for a very important reason. It helps to prevent them from lysing when they're in a hypotonic environment. What do you mean by hypotonic?
No water,
It means high water, low solute.
no
As I said before, and I'll say it again, water follows the solute. So a hypotonic environment, if a cell is there, and they're in a hypotonic environment, that means there's more water outside the cell than inside, so water is naturally going to flow into the cell. Okay? If the cell has a rigid cell wall, it can resist the buildup of pressure as that water comes in, like many bacteria. So the bacteria has a damaged or destroyed cell wall. As that water comes in, boom, it blows the cell apart, lyses it, kills the cell. So that can be very important as far as an antimicrobial effect. Lysozyme. Okay, we also have, of course, the wax in the ears, that's cerumen, and that helps to keep things from going deeper into the ears because it traps a lot of debris. So this first line of defense consists of the barriers, and the point is to keep down or prevent organisms from getting in the body in the first place. It doesn't keep everything out, but it often reduces the numbers of organisms that can get into the body. Also, as you know, the gastric juice. The gastric juice is what's secreted by cells in our stomach wall. We'll talk about that more when we get into the digestive system. But as you know, it's acidic. And it's actually pretty low pH. It can be from about 1.2 up to about 3, so it's very acidic. You can literally dissolve metal in your stomach. Don't try it at home. It's very hard getting that metal down. But anyway, it's very acidic. And why is it so acidic? Because we secrete hydrochloric acid, very concentrated hydrochloric acid. The purpose, of course, is to kill microorganisms. So the stomach is a very hostile environment for most microorganisms. What many organisms do is they come in with the food, and they're cloaked in the food, and then they slip into the small intestine. They're more likely to survive there than they are in the stomach. Although there are a few organisms that can live in the stomach. Okay, so that gastric juice, again, helps to kill many of the microorganisms coming into the body as a result. It also does some other things, too, which we'll talk about later. And as you know, you got an upset stomach and you regurgitate, okay, you may feel better, but you got this burning sensation in your throat. That's because the acidity in that gastric juice is burning the lining of the esophagus. And sometimes in indigestion you get heartburn, which has nothing to do with the heart, but what you're doing is you're getting some of that stomach acid up, okay, irritating or burning the lining of the esophagus, hence the term, because it's near the heart, okay. Now they got a fancy name for it, it's called it's called GERD, gastroesophageal reflux disease. And of course there are pills for that too.
Of course.
You know, I love that one with the guy. He's walking there. He's got flames coming all over. You know, do I take the pill now and then eat, or do I eat and then take the pill now? What are you eating it for? You know, in the first place. You know, you shouldn't be doing this. You know, if something upsets your stomach, don't eat it. Don't need to take all these fancy pills. Okay, so that's part of the first line of defense. The barriers, skin and mucous membranes, and various chemicals associated with them to help keep microorganisms getting the body in the first place, but failing that, reducing the numbers. Now, the second line of defense is also non-specific. This doesn't consist of barriers per se, but it's cells and various processes associated with these cells. And what this does is deal with organisms that have gotten into the body. Because you're always going to get some organisms in. Like I say, you're constantly being bombarded by microorganisms. Every time you put something in your mouth, you rub your eyes, your fingers up the nose, whatever, you're inoculating yourself with microorganisms. And of course, we don't even think about it, you throw a finger, you stick a pencil or pen in your mouth, or whatever. So you're going to have to deal with microorganisms that have gotten in the body. So one part of the second line of defense is a process called phagocytosis. We mentioned this before. There are cells that are called phagocytes that engage in phagocytosis. So the cells that carry on phagocytosis are called phagocytes, and there are certain types of white blood cells that are phagocytes. And again, we mentioned this before, we were talking about the white blood cells, but leukocytosis, that's an increase in number of white blood cells, and a decrease leukopenia. And either can indicate infection. Frequently, an infection is associated with an elevated white cell count, leukocytosis. I just listed a few diseases here. Appendicitis, we get inflammation of the appendix and intestine, gonorrhea, which is sexually transmitted disease, infectious mononucleosis, just to name a few. And in fact, in most infections you're going to see an elevated white cell count, leukocytosis. But in some, you actually have a reduction of white cell count, because the parasite may kill the white blood cells, or that causes brucellosis, for example, that which causes pertussis, better known as whooping cough, salmonellosis, which causes all sorts of infections, especially GI tract infections, and some of the viral infections that we get, and some of the so-called rickettsial infections. Rickettsia are very tiny bacteria, and like viruses, they're obligate intracellular parasites, which is to say they can't reproduce on their own outside the cells. They have to get into a cell in order to replicate. So they're dependent on living cells. And all viruses, of course, are not even cellular. They have no metabolism, and what they do is they come into a host cell and basically degrade the host cell DNA and then use their DNA, or in some cases RNA, to take control of the cell, and use the cell then as a virus-producing vacuum. And in the process, of course, you just destroy the cell. And again, leukopenia can result because this may interfere with leukocyte production by breaking up their membrane. And like I say, some of these infections actually result in the destruction of white blood cells. So white blood cells don't always kill the parasites. Sometimes the parasite gets the upper hand. Okay, and the phagocytic cells, of course, are the neutrophils. I'm sure you realize by now. And of course, this is the most numerous of the white blood cells, which you've seen from looking at these blood cells. And usually, this is the first cell you see, producing great numbers early in an infection. And they're usually first on the scene. When you've got an infection in the site, the neutrophils usually are there very quickly and in large numbers. And then as the infection progresses, eventually subsides, then we begin to see more monocytes predominating, and they also will be migrated. Of course, when they get into the tissues from the blood, they become macrophages. And these two, like the neutrophils, of course, are phagocytic. Some of these macrophages move around in the tissues. They're called wandering macrophages. Some reside in a particular organ. They're called fixed macrophages, and usually have a fancy name associated with them. They all do the same thing. They encounter, ingest, and usually digest solid particulate matter they take in. As we're going to see, they're also important in helping to activate lymphocytes. They're called antigen-presenting cells, which we'll discuss later. Because that's what they literally do. They present foreign antigens to lymphocytes to wake them up, activate them. called fixed. Macrophages are also called histiocytes, which basically means a cell in the tissues. Histology is the study of tissue. Cyte means cell. All right, in the liver, they're called the Kupffer cells. We've already seen what things they do in the liver. Well, maybe we haven't seen it. Well, we will. I'm ahead of myself. The liver, like the spleen, also destroys worn-out red blood cells, which is why you can live without a spleen, not a liver, but a spleen. We've got them also in the lungs. Here they're called alveolar macrophages, and they migrate around the respiratory passages, especially the tiny little air sacs where most of the gas exchange occurs in the blood. Those are called alveoli. They're called alveolar macrophages. In the nervous system, these are mitochondria. Remember some of the glial cells. We'll probably talk about the different kinds of glial cells in AVI, 6 different kinds, 4 in the CNS, central nervous system, and then 2 in the PNS, peripheral nervous system. You know all about those. And then in the epidermis of skin, we got the Langerhans cells. We've also got these in the bronchial tissue, we've got them in the bone marrow, we've got them in the lymph nodes, of course, spleen, and also one of them up in the peritoneal cavity as well. As I said too, you already know that the macrophages destroy debris and red blood cells in the spleen, but this also occurs in the liver. Again, by virtue of these macrophages sitting close together. So let's take a quick look at the process of phagocytosis, as described in your book. So the first thing, of course, is the phagocyte has to find a parasite. And usually, the parasites are pathogens that get into the body are causing tissue damage, and the result is the release of chemicals. And phagocytes are attracted to these areas of infection by following a chemical trail. And that's referred to as chemotaxis, movement in response to a chemical. Positive chemotaxis.
It's kind of like chemotaxi.
Yeah, if you will.
Takes it through.
Chemo, chemical. Right.
Taxi.
The taxi is usually, of course, the blood system following. They're following this chemical trail. And what chemicals are we talking about? Well, we're talking about chemicals that are released from the damaged host cells. We're talking about chemicals that are released by the microorganism itself when it's metabolizing. And also chemicals released by the leukocytes that are already there. They bring in more. They bring in reinforcements. They can track one another. In fact, that's how cells communicate with one another. They don't talk. They communicate with chemicals. They release chemicals, and cells that respond to those chemicals have specific receptors on their cell membranes, which are complementary. And we see that's very important in the specific response between lymphocytes to communicate with one another by chemicals being released and chemicals being picked up. Okay, so chemotaxis. This is how they know where to go. They're following this chemical trail. Again, the chemicals can be chemicals released from the damaged host cells, chemicals released by the metabolism of the parasites, the bacteria, whatever, are there, and then chemicals released by white blood cells that are already in the area. Now, once they get there, they have to be able to attach to the organism. So it's to adhere to them, to stick them. So the cytoplasmic membrane is going to attach to the microorganism. Now, some microorganisms will resist phagocytosis. Some bacteria, for example, have what's called a capsule surrounding their cell wall. And the capsule is kind of gelatin-like. It's full of water. And it's very slippery, and it can make it very difficult for a phagocyte to grab hold, basically, on the microorganism. And in fact, in some organisms, some bacteria that are pathogens, the strains that are pathogenic have a capsule, and the strains that are not pathogenic, don't cause disease, don't have a capsule. So they're ready to phagocytize and destroy. A good example would be pneumococcus pneumoniae, which is a common organism that causes pneumonia. particularly dangerous in the elderly. The encapsulated strains are the pathogenic ones, and the non-encapsulated strains are readily destroyed by the host phagocytes and destroyed, so they don't cause disease. This isn't to say that because an organism has a capsid, it's going to be a pathogen or disease causer, but it can contribute to the virulence or severity of the infection if the organism does have this capsid. And the capsule depends on the bacterium, it can be polysaccharide, polypeptide, whatever, but it's slippery. Okay? So in some cases, the phagocyte can't gain hold unless it can push it against something, like the wall of a blood vessel or some fiber or something like that. However, phagocytes are added, or aided rather, when that parasite cell becomes coated with protein. And those proteins are called opsonins. Now the proteins may be plasma proteins, such as complement proteins, which we'll talk about later, or they may actually be antibodies. Well, once that cell is usually coated with these proteins, it's normally readily phagocytized and destroyed. And again, these proteins that coat the cell are called opsonins, the process is called opsonization, which we'll get to. All right, once they gain hold of the cell, okay, they found it, they attach to it, then what they do is throw off cytoplasmic extensions, like an amoeba, completely envelop that cell, and basically form a little bag around the cell made up of the membrane of that phagocyte. That part then, detached from the rest of the phagocyte membrane, comes to the cytoplasm of the phagocyte, and this is called a phagocytic vesicle. But again, the walls of that vesicle are originally part of the phagocyte's membrane. And then what usually happens is, excuse me, the phagocytes lysosomes. And remember, most cells have lysosomes. What are lysosomes?
They cut it up.
Bags of digestive enzymes, okay, that are used for intracellular digestion. These phagocytes lysosomes then will fuse with a phagocytic vesicle that contains the organism. It'll discharge its digestive enzymes into that. Now it's called a phagolysosome. And that usually then will digest the organism. Now some organisms can resist this. They may be taken in, and they resist this, and they can kill. Sometimes they can kill the phagocytes by toxins they produce, like some Staphylococcus species, for example. Mycobacterium tuberculosis, which is the organism that causes tuberculosis. It's very resistant to phagocytosis. They can actually multiply inside the phagocytes and eventually kill them. So, phagocyte isn't always successful in killing organisms. Sometimes the other way around. The organism that causes these known as tularemia, which is Francisella tularensis, and also one that causes brucellosis, that's a Brucella species. These can actually get into the phagocyte and just remain in a dormant state, and in doing so, they're protected from antibodies. They're sort of hiding out there. As we're going to see, the phagocytes are also important. Some of these phagocytes are important in specific defenses because they aid in activation of lymphocytes, both B and T lymphocytes, which we'll discuss later and look at specific immunity. This is from your textbook, and it just shows the events in phagocytosis. This represents a phagocyte. Okay, and this is the pathogen or the parasite. And you can see what happens is, of course, once the phagocyte comes in contact with that parasite, it basically envelops it. It throws off cytoplasmic extensions completely around that cell. And then that part of the membrane, which contains the pathogen, attached to the rest of the membrane, comes into the cytoplasm. And that's the phagocytic vesicle. That then will attach to the cell's lysosomes. They'll form this phagolysosome, and of course then the digestive enzymes, the hydrolytic enzymes, go into the vesicle, and this usually then will digest the organism. And then the remnants, of course, are simply expelled, the waste products. cells that do this, of course, are called phagocytes. So we have neutrophils that are phagocytes. We have the monocytes, which of course become macrophages when they leave the blood, and eosinophils. Any questions on this? Okay, and again, the phagocytes are part of the second line of defense. They deal with organisms that have gotten into the body. Also part of the second line of defense, another type of cell. Now, these are not phagocytic. These are called natural killer cells or natural killer lymphocytes. Remember we talked about lymphocytes, and I said most lymphocytes are small, not much bigger than a red blood cell. There's some large lymphocytes. These are large lymphocytes. They're not as common as a typical lymphocyte. They're distinctly larger. They do have some granules in their cytoplasm. Okay? And what they do is they kill other cells, hence the name natural killer cells, or NK cells for short, or sometimes natural killer lymphocytes. So they're not your typical lymphocyte. Because unlike most lymphocytes, they are non-specific. What do lymphocytes do? What's their function? What did I tell you? Specific immunity. These are not specific. They will attack and attempt to destroy anything that is foreign. And by that I mean abnormal cells. And these are cells, body cells, are infected with a parasite, like a virus, or a bacterium that's gotten into the cell, or cells that become transformed into cancer or tumor cells. They attack and destroy these cells, not by phagocytosis, but by releasing toxic chemicals. So they are killer cells, yes.
Does that do the same thing as RA,
That's what?
as rheumatoid arthritis? Is that something that,
That's what?
rheumatoid arthritis?
Well, rheumatoid arthritis, you've got your own cells attacking the tissue. And usually the cells that do this would be like the C cells, the T lymphocytes, which are very specific. We may play a minor role, but in most cases, your T lymphocytes. But notice, these are not immunologically specific, which means they don't seek out any particular organism, just anything foreign. And often it's a host cell that has become parasitized. How does it know? How does the immune system know that that cell is parasitized? Because it often has antigens posted on the surface that are different than the normal antigens. So the immune system has recognized this is a cell that's different. And being different means destruction. You want to destroy it. And these cells take part in that process. But again, unlike the typical lymphocyte, B and T lymphocytes, which are very specific, respond to a specific antigen, a foreign antigen, these are not. Just anything foreign. And usually it's a body cell that has somehow become transformed because it's got a parasite inside of it, or it's become a tumor cell. Because all our tumor cells, all our cancer cells, are derived from normal body cells that have somehow become changed. Now again, our NK cells are natural killer cells. Some other cells are involved in non-specific defense. This would include the macrophages, the neutrophils, the eosinophils. And they will readily kill cells that are coated, as I said, with protein, such as antibodies. And they basically have receptors that are specific for part of the antibody molecule, which we'll talk about later, what's called the constant region. And you're going to see what they do, basically. Antibodies are usually described as being kind of a Y-shaped molecule. Like this. And they have a variable region up here where they attach the antigen, and then down here is the so-called constant region. I'll go through this again later. We get to talk about antibody-specific response. Okay? But these will attach to, like the cell, okay? And this will attach to the other cells, like the macrophage, neutrophils, eosinophils, the NK cells, will attach to the stem. And what does that do? It allows it to grab hold, adhere to that foreign cell, and thus heal it, phagocytize it, or kill it with chemicals. Okay? And again, I'll go through this when we get to the specific immune system. Okay, so we talked about the second line of defense. We've talked about phagocytic cells that ingest and digest, destroy cells for the most part. We've talked about the natural killer cells, which don't kill by phagocytosis, but by releasing toxic chemicals. And now also part of the second line of defense is a process called inflammation. You've all had inflammation one time in your life. You get a cut, okay, what happens? It bleeds for a while, it stops bleeding, and then it becomes inflamed. Meaning what? The skin becomes darker or reddened. It's warmer, okay? It swells, and it hurts. These are typical signs and symptoms of inflammation. That's part of our second line of defense. Why inflammation? The purpose is to destroy the invading organism or any toxic products it produces. Because in some cases, the disease is not due to the organism per se, it's due to toxic products it produces. A toxin, for example. Tetanus, a good example. You've all heard of tetanus. Tetanus is a disease caused by a very potent neurotoxin released by the organism. What it does is interfere with nerve transmission to the muscles. And it results in freezing your skeletal muscles in a permanent contractile state. It usually starts up here in the jaw, and that's why another name for tetanus is lockjaw, because it freezes those muscles of mastication. Then it goes down to the neck. You have trouble swallowing and speaking. It can paralyze those muscles as well. Then it starts to spread out to other muscles in the body. Think of the worst cramp you've ever had in your life and translate to the major muscle of your body. That's what the stages of tetanus are like. And tetanus untreated is fatal. When it gets to respiratory muscles, you die of respiratory paralysis. And in fact, the muscle contracture is so severe in the terminal stage of tetanus, it breaks bones. And the spine basically just goes into a bow shape, and the body's just rigid. Horrible. Nasty disease. And there's no reason to get it because we have a very effective vaccine for it. And you don't get the disease by stepping on a rusty nail. Okay? It may be a bright, shiny nail, but if it's been exposed to the soil, and that soil contains the endospores of the parasite, and you get that in the wound, you just inject yourself with the organism, it produces the endotoxin, and you end up with tetanus as a result. If you've had the tetanus shot, you've got antibodies against it, that inactivates the toxin, you don't get the disease.
Isn't that only good for 5 years though?
Well, they're saying like about every 5 years or so. Okay? And if you can't remember the last time you had a tetanus shot, you have some wound, they'll usually give you one. Okay? And some people say, well, I have such a reaction to it. Well, I'll tell you, the reaction to the shot is much less than getting tetanus. Okay? You do not want to get tetanus. And it's around here. It's all over the place. The organism is a soil-dwelling organism. So contamination of that soil getting into a wound